Perfect.
Okay, thank you very much for having me. We have a small shift in people in subject study. Now I would like to talk to you not about animals, but about humans, and in particular about manipulating the dopaminergic midbrain in the lab. As you know, the dopamine-producing neurons, they are located primarily in two areas in the midbrain, the ventral tegmental area and the substantia nigra. And they project to the striatum primarily and to the prefrontal cortex. And because the dopaminergic system is involved in a variety of disorders such as Parkinson's disease or addiction or schizophrenia, the question has come up whether it would be possible to manipulate it, and of course it would be desirable to manipulate it in a relatively physiological way. Multiple methods are available for manipulating dopamine systems. One of them is to take advantage of the cortical input onto the striatum, where cortical input comes together on spiny neurons with dopaminergic input from the midbrain, and use TMS. So in TMS, basically, one can, for example, stimulate motor cortex and then show with PET that this releases dopamine in the striatum. But I'm not going to talk more about this, just for to mention that this exists as well. And then we can also, or sort of the standard method, of course, to manipulate the dopamine system is to use pharmacological interventions. And using these interventions, many people here, including Mathias, of course, have shown that the dopaminergic system plays a causal role for value-based decision-making, including social preferences, intertemporal preferences, and risk preferences. We ourselves also have used this method, and for example, we have shown that you can affect effort discounting. So we have used, in this case, a D1 agonist in humans, and basically showed that with increasing dose of this D1 agonist, people are more willing to exert effort. But in the same time, when we use pharmacological manipulations, we have issues like this. So we had a substantial number of people showing side effects. So particularly in this case, nausea, but and it was quite specific to the higher doses. And so that basically highlights one of the limitations of pharmacological manipulations, which is that it has side effects. Another one is that the time course of many of these pharmacological interventions is quite slow. So for example, amisulpride, which is another one typically used to manipulate the dopamine system, a D2 antagonist, has basically a half-life of about 12 hours. And the question then basically is whether these changes in dopaminergic concentrations or dopaminergic action are still physiological, or whether we are sort of looking at something slightly different than the neural computations that are supposed to be ongoing and be performed by dopamine neurons underneath. And one possible intervention then, or manipulation, may come from real-time fMRI or neurofeedback used in combination with self-regulation of the dopaminergic midbrain. So the idea there, you probably know, is you have a region of interest where, from which you measure activity. You analyze the activity basically in real time. You display it to the participants, and they try with some strategy to upregulate or downregulate the activity. And then this again, you measure the activity and so on. And so in this case now, we would like to manipulate or self-regulate the substantia nigra and ventral segmental area. And one obvious strategy then that one may want to think of is reward imagination. So as you all know, substantia nigra and VTA neurons at least have been implicated in reward processing. And when participants are lying in the scanner, of course, they need some sort of strategy or some sort of idea of what they should do in order to upregulate or downregulate activity from a particular region. And one strategy that has been used, and I'm going to show you later, is reward imagination. And it actually does make sense in terms of at least simple fold results that we know of so far, in that it has been shown that simply having conditioned cues predicting imagined reward elicit activation in the dopaminergic midbrain, or at least in the midbrain. And so you can see here basically increased activity to cues associated with reward imagination compared to those cues associated with no reward, and as well somewhat increased activity to cues predicting visually presented reward. And also in a separate experiment, it has been shown that also increasing magnitude of hypothetical reward also activates the midbrain more strongly. So that basically gives us slightly more confidence that in principle a reward imagination strategy should be possible for neurofeedback. And exactly this has been used. So here I'm showing you an example. I'm going to show you the actual task slightly later, where people were asked in 3 runs in this case. In some blocks, they were asked to upregulate the substantia nigra VTA or rest. And basically what we see is the increase or the activity that is measured during these neurofeedback training runs, as well as during baseline and transfer. So baseline, there is no activity displayed on the screen. People just are told, please upregulate the activity that you're supposed to upregulate. And of course, during baseline, they have no clue what they're supposed to do. But then during transfer, they're asked to do this again, or they're asked to upregulate again. But now, basically, they're supposed to be able to transfer what they have learned during the training runs, right? So now, without having visual feedback, the hope is that people are able to upregulate substantia nigra and VTA. And in this previous study here by James Sulzer and colleagues, they basically had 2 groups. One group in which the feedback was veridical, so whenever activity went up, the signal also went up that was displayed on the screen. And another group where the feedback signal was inverted, so whenever the activity went up, the signal went down. And what they basically find is that in at least the second run of these 3 training runs, there is a significant difference between the 2 groups, but also they find relatively little transfer. Second study, and this was with reward imagination. So basically people were asked to imagine things that make them happy, so romantic images or motivational achievements, so winning a race or passing time with their loved ones, good food, and so on. And a second study has used exactly the same strategy, this time only with 2 runs, but otherwise everything else was the same. And not only in healthy controls, but also in cocaine users. And again, they find that during training, people are able to upregulate, but again, there's relatively little transfer in both groups. So it seems to be the case that one can learn, at least as long as the feedback is present, to upregulate the midbrain, but the transfer seems to be relatively unstable, or at least there seems to be a lot of variation in transfer. So we then asked whether we can use these same two existing data sets that I just showed you to shed a bit of light on how transfer comes about. And what we also ask is basically whether individuals who show more transfer also differ from individuals who show less transfer. And this is the task that both of these studies have used. Basically, during baseline and transfer, people are just instructed to either rest or imagine positive things, right? And there is no feedback, no activity shown on the screen. And then during the actual training, they had, again, also blocks of rest and upregulation, but this time they viewed the activity from the SNVTA here. And this activity was, they were basically supposed to upregulate it, both in the standard feedback group and in the inverted feedback group. Okay? So what was, but I've already shown you, what was clear from already the published data was that when you look at transfer, so when you take the difference between happy time and rest and compare it between transfer and baseline, on average, there is little effect of the neurofeedback training, right? So people on average don't show much of an increase. But as you can see as well, there is quite a lot of individual variation. So some people do manage to increase, whereas others do not, or even get worse. Okay, and so as a manipulation check, we first wanted to know whether those who actually do learn to increase, whether they actually also show stronger increase during training. So we compared, or we made a correlation basically between the transfer effect, so transfer minus baseline, and the increase in activity for happy time minus rest during training. And in the vertical feedback group, we saw a relatively strong relation. So people who were better at transfer showed also a stronger increase during training. But that was not the case in the inverted feedback group. Okay, so then we asked whether there would be regions outside the SNVTA which show a relation with transfer success. So we basically correlated the midbrain transfer, the individual degree of midbrain transfer, with the rest of the brain. And we found primarily a region in cortical control regions, such as the dorsolateral prefrontal cortex and the ACC, showing showing a correlation with the degree of midbrain transfer. So basically, the more people were able to regulate during transfer, the stronger these regions co-activated also during transfer. And in control analysis, we checked whether this was indeed specific to the substantia nigra VTA. We used an ROI close by in the parahippocampus and found that we didn't find these same regions correlating with changes in the parahippocampus transfer. And also we found that it was specific again for the vertical feedback group. So next we asked whether changes during learning itself, during neurofeedback training, also correlate with transfer success. And basically what we did now was not to analyze data on a blockwise fashion, but in a TR by TR fashion. And the idea basically here is that people try to learn continuously whether their strategies work or not, right, during neurofeedback training. And in sort of a prediction framework, they predict the feedback of the next TR based on what they're doing currently. And then the next TR elicits, in this framework at least, a prediction error. So either there is higher activity than they expected or lower activity than they expected, given the current activity and what they do currently, or no change in activity. And so basically, we just took a trial by trial or TR by TR resolved prediction error signal, prediction error-like signal, by taking the difference between TR at time point I and time point I minus 1, right? So successive time points. And we reasoned that the degree of prediction error coding should decrease more in people who learn more, right? So it becomes less, and you should be less and less surprised as you learn more and more about the efficacy of your own strategy. And this indeed is what we find. So people who learn who show stronger transfer, they also show stronger decrease in prediction error coding over training. So transfer correlates with neurofeedback training decrease of this prediction error-like signal in dorsolateral prefrontal cortex again. Then we used this region that we just identified with the prediction error coding analysis for functional connectivity analysis, and basically used this dorsolateral prefrontal region as a seed and asked whether the connectivity of this region changes with SNVTA as a function of midbrain transfer success. And again, we found a correlation such that the more successful people were in transferring activity, activation regulation, the more the connectivity between these two regions increased. Finally, we asked whether individual differences in reward sensitivity also discriminated between people who were more or less successful in regulation transfer. And to do that, we have basically used a separate task, which was performed by a subset of people in the second study, namely a monetary incentive delay task, where people viewed cues which predicted different magnitudes of reward. This one cue here predicted between 0 and 2, another cue predicted between 0 and 0.4 francs, and the third cue predicted no reward at all. And then they basically had to respond as quickly as possible, and if they responded correctly and were fast, they received or earned more money in the current trial. And the idea then is that with these two conditions here, the high reward and the low reward, we have different standard deviations or spreads of reward outcomes. And in an adaptive coding framework, one would expect that humans, or at least, or also dopamine neurons show this kind of behavior, that a neural system that is adapting to the most likely outcome distribution shows a steeper increase in activity for the small reward range than the large reward range, and thereby uses its neural machinery most efficiently for the most likely rewards. And so basically, we looked at overall reward sensitivity, basically the sum of the parametric modulators of these two, as well as for the difference, which we would then interpret as adaptive coding. And again,
Philippe, can you hear me?
I'm almost done.
Yeah,
sorry.
You have 1 minute if you want to give some.
So basically, just to conclude, we find again a correlation in lateral prefrontal cortex with reward sensitivity, both adaptive coding and reward sensitivity. And basically, lateral prefrontal cortex seems to indeed characterize people more strongly who show stronger regulation transfer success. So I think it's still a fair while until we have this as a fully established third method, but at least I hope to have shown you some steps. Sorry for running over.
No, that's fine. Thank you, Philippe. Super interesting, indeed. Maybe one short question. So the main contrast you have between happy and rest seems to confirm two things, right? There's the valence of the state. So one is happy or positive, and the other is neutral. And then there's a task. In one case, you're trying to imagine something. In the other case, you're just mind wandering, I guess.
Sorry, I should have said that. People were also supposed to do something.
Okay.
Yeah, in the rest condition.
Okay. But my question was, would it be possible to get a better contrast between happy and unhappy or sad kind of episodes you would imagine in your mind? Or
yes, in principle, yes, but you would increase the difference between the two, but at the same time, you would not be able to say whether it's specific to reward, right? It could also, so it's part of both then.
Mm-hmm.
Right? You're combining positive and aversive.
I see. Do we have other questions for Philippe? I can't see any. Yeah, so we may get them at the end. So in the meantime,